Greetings. We continue our lessons in terms of learning the metabolic pathways. This is ideally after we have already learned about all the macromolecules, that is starting with nucleic acid. Then we went through in terms of protein, as well as one of the topics very much relevant for metabolic pathway, that is enzyme. And then we followed suit in terms of carbohydrate. And lastly, we looked at lipid. So we start our journey in terms of learning about metabolic pathway by starting with glycolysis. To me, glycolysis or glycolytic pathway, I consider it as the major catabolic pathway. Why? Because it is part of the glycolytic followed by citric acid cycle and then followed by electron transport chain. The cell actually utilizes these pathways in order to obtain ATP, and the term catabolic refers to from a big molecule such as glucose become oxidized until become carbon dioxide as well as water. So the journey for glucose in the cell is actually starting with glycolytic pathway, where it will become from 6 carbon to something of a 3 carbon pyruvate. So glycolysis, or also known as Embden-Meyerhof pathway, it is in honor of these two individuals, that is Gustav Embden as well as Otto Meyerhof, who have spent time to discover this pathway. They didn't invent it. You can't say they invented this pathway, because the pathway is all along present in the cell. It's a matter of discovering the pathway. And in defining this pathway, it is referred as either taking glucose and oxidizing it to become pyruvate when there is ample of oxygen, and also having organelles such as mitochondria in a particular cell. You will see when there is no mitochondria, it will end up to become lactate. That means it starts with glucose and become lactate because of no oxygen or lack of oxygen. So that's where, in terms of the site where this glycolytic pathway takes place, it is commonly understood it will take place in cytoplasma. But in the body, there are tissues, there are organs. To differentiate between certain tissues or organs, you will see that, such as erythrocyte or red blood cell, the mature red blood cell is missing a few organelles such as mitochondria as well as nucleus. Why? Because it can be much more flexible. The biconcave shape is much more flexible to go through capillaries when it has less organelles. However, since it does not have mitochondria, therefore it cannot do citric acid cycle as well as electron transport chain. So what happens in red blood cell, it only stops up to glycolysis and glucose will become lactic acid. So because there's no mitochondria. Then you have something of that number 3 shown there. These are muscle cells, muscle or myocytes, vigorously taking part in oxidizing glucose, especially during exercise or especially during sports. So in that conditions, the lack of oxygen takes place, and therefore it shifts from complete oxidation of glucose to somewhat partial oxidation, and that's where glucose is converted to lactic acid instead of being pyruvate, and that's been shown there in number 3. And then, of course, in between, there are also tissues that is lacking of mitochondria, not enough. So they have both, but not in relying on one most of the time. So usually in my lecture, I put this kind of slides with YouTube videos to make you understand more about the pathway itself. Basically, it's just to make it interesting because metabolic pathway can tend to be very monotonous, and therefore it'll be good to have this sort of videos for you to understand better. In this video, there are two types of individual, one being a sprinter, the other one being a marathoner. The sprinter will have more, stocky build-up with muscle because he needs to use the muscle more, while the marathoner less of a muscle build-up but more of a lean body with less fat. So he relies more on aerobic kind of glycolytic pathway rather than being anaerobic. While the sprinter, although he may start off with aerobic before reaching the 100-meter dash, he tend to be in anaerobic most of the time because lack of oxygen, because wanting to achieve fast sprint. While the marathoner can tend to be utilizing the fat as energy source, so he tend to be also aerobic as well as anaerobic. So since this is the first metabolic pathway that you are being exposed to within the course of biochemistry, just for your understanding, normally the pathway lectures will be looking into every step of the pathway, the chemical or the biochemical reaction step by step. And also the metabolic pathway tend to be in phases or stages. So in that context, you can see that glycolytic pathway is actually divided into two stages, that is stage 1 as well as stage 2. The stage 1 is somewhat need to invest energy, so the system need to use energy, and that energy comes in the form of ATP hydrolysis. So ATP, in short, ATP need to be utilized here. In fact, there will be two ATP molecules to be hydrolyzed in 1 to 5 steps of this stage 1. Okay. Glycolytic pathway, total of 10 steps, and these are the first 5 steps where 6 carbon will become 3 carbon glyceraldehyde 3-phosphate. Therefore, in some books, biochemistry book, they also call this as hexose stage. And then you have stage 2, where energy actually return back. That means ATP is produced, and there will be a total of 4 ATP being produced at the stage 2, or also known as triose stage, because all these steps 6 to 10 of glycolytic pathway is actually involving 3 carbon intermediates. Okay, and there's also shown there the N ATP production because of the 10 steps of glycolysis will be 4 minus 2, that is net of 2 ATPs. So when glucose undergoes oxidation from glucose up to 2 molecules of pyruvate, the total number of ATP produced will be 2 net ATP, whereby at stage 1 need to invest 2 ATP, and at stage 2 it will produce 4 ATP. So that's why I got 2 net ATP. So here gives you in terms of location. Another thing about biochemical pathway, it's important to know where the location of a metabolic pathway takes place. In terms of glycolysis, You can see there that glycolysis takes place in the cytoplasma of a cell, and whereby glucose will undergo oxidation with 10 steps, and the end product will be pyruvic acid or pyruvate, and it will be 2 pyruvate that will be obtained, because glucose is 6 carbon, pyruvate or pyruvic acid is 3 carbon. And then what happens is the pyruvic acid will go into the mitochondria, will go through Krebs cycle or citric acid cycle. There will be around 8 steps, and eventually within the Krebs cycle there will be ATP produced, as well as NADH, as well as FADH2. These are all considered reduced coenzymes. In glycolytic pathway also, there will be 2 NADH produced at a certain step of the 10 steps reaction. And these NADH, FADH2 will then go into electron transport chain. So if glycolytic pathway got 2 NADH, it has to go into the mitochondria, go into the electron transport chain pathway in order to be oxidized. Just now it was reduced, now it will be oxidized, and when it's oxidized, it will then generate NAD, NAD plus or FAD, and in the process also generate ATP through what is known as oxidative phosphorylation, but that one we will learn in the lecture of electron transport chain. Just want you to understand, apart from oxidative phosphorylation, which happens in the electron transport chain, there is also something known as substrate level phosphorylation, and this happens in a step in the glycolytic pathway, in the steps in the glycolytic pathway, as well as the Krebs cycle. What's the meaning of substrate level phosphorylation? Within the reaction steps, high energy phosphate will be given to ADP, or in the Krebs cycle, GDP, guanosine diphosphate or adenine diphosphate. When the high energy phosphate is given by the intermediate, the ADP will become ATP. That means do not need the electron transport chain to build ATP, but within the glycolytic pathway steps or Krebs cycle can generate ATP or GTP. So another thing about learning about biochemical pathway, this is the first pathway. There are many more pathways to learn. There is also something about the steps. Every steps you need to know changes, but for the effect of this course, I am not requiring you to memorize every single step, not requiring you to memorize every changes happening, for instance, to the glucose, not expecting you to know or memorize or draw the structure in terms of what changes happening to the structure, knowing in detail all the enzyme that is involved in the metabolic pathway. When it comes to the glycolytic pathway, I told you there's 2 stages. This is the stage of energy investment phase, or hexose stage. It has 5 steps, therefore 5 enzymes, and it starts off with glucose. There you have glucose, and then this is where ATP is hydrolyzed, and the phosphate will be given to the glucose to become glucose 6-phosphate. As you can see, it's added with glucose 6 to become glucose 6-phosphate, And this is done by the enzyme hexokinase. Next, the glucose 6-phosphate creates an isomer in the form of fructose 6-phosphate. Naturally, you require an isomerase. Then there is another energy investment done through the form of ATP, where ATP hydrolysis takes place due to this enzyme, and therefore you will have phosphate now in both sides of the fructose. Therefore, this is known as fructose 1,6-bisphosphate. What happened next due to the aldolase is there will be a cut or cleavage, and therefore you will get from 6 carbon to become 3 carbon dihydroxyphosphate as well as glyceraldehyde 3-phosphate. Okay. But the dihydroxyacetone phosphate, this one, does not stay as dihydroxyacetone phosphate. It will be converted to glyceraldehyde 3-phosphate because of the effect of isomerase. So what started with 6 carbon glucose will then end at the first stage to become 2 molecules of glyceraldehyde 3-phosphate. Before I go to the next slide, I just want to tell you metabolic pathway have enzymes, but they have crucial enzymes which are considered as key regulatory enzyme. These enzymes are the one important in playing the role of regulating the whole pathway. In glycolytic or glycolysis, there are 3 key regulatory enzymes, and 2 of it is being shown here. That is hexokinase as well as phosphofructokinase. Later we will see there is also one more enzyme, that is pyruvate kinase. So these are the 3 most important enzymes in glycolysis, whereby if you can control this enzyme, you can either activate this pathway or inhibit the whole pathway. So to continue on going in detail about glycolytic pathway based on stages, now we look at what happens on the second stage, which is also referred as energy payoff phase, which has 5 more steps, and it is also referred as triose stage because all the steps of 6 to 10 involves 3 carbon. So we already know that there are 2 glyceraldehyde 3-phosphate intermediate will now enter the energy payoff phase. And what happens is, first there will be buildup of 2 NADH, and this is important because it will then go to electron transport chain, where it can then generate for each NADH 3 ATP. Okay? So here you get 2 NADH, and then you will, from glyceraldehyde 3-phosphate, you will get 1,3-bisphosphoglycerate. Why? Because the glyceraldehyde 3-phosphate will now have attachment of phosphate here as well as here. And then this number, don't forget, because you have 2 glyceraldehyde 3-phosphate, therefore you get 2 times 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate is having high energy phosphate. That phosphate will be given directly to the ADP, 2 ADPs, to become 2 ATP. So now, once energy has been invested, now the energy is pay off. Therefore, you get 2 ATP. And then 1,3-bisphosphoglycerate, it will become 2-phosphoglycerate because it loses 1 high energy phosphate. Next, what happened is some rearrangement. So 3-phosphoglycerate now will become 2-phosphoglycerate because the phosphate changes location. And due to this enzyme enolase, hydro removal of water molecule will take place, dehydration, and therefore there is also formation of double bond there. And lastly, because of pyruvate kinase, you will get this phosphate with high energy in it will be transferred to ADP, and finally that will make into ATP, so 2 ATP. So if you look back, you have obtained 4 ATPs within this energy payoff phase. Then if you take into account the loss of the ATP, there's 2 ATPs lost in the first stage. So what's the net ATP? 2 ATPs in net. And then don't forget, you also have 2 NADH. And in terms of this slide, lastly, I also want to emphasize, remember I told you there are 3 key regulatory enzymes. The other enzyme which is important for glycolysis pathway is pyruvate kinase. Next, we look at into what happens to glucose, or rather what happens to the pyruvate once it has oxygen, ample oxygen, or when there is also mitochondria is present in that cell. So under these two conditions, the pyruvate that is formed by glycolytic pathway of glucose will carry on until become carbon dioxide and water. And in the process, it will also form NADH, FADH2, because in the Krebs cycle, it will form both of these NADH as well as FADH2. So that's the effect of pyruvate. It doesn't stop at being pyruvate. It carries on into the mitochondria and become eventually carbon dioxide and water because ample supply of oxygen is present. So here shown what I just explained in the previous slide in terms of what happens to the fate of glucose, or rather the pyruvate, once it has completed glycolysis. You can see that the glucose, okay, within the cytoplasma, it will end up being pyruvate using 10 steps of glycolytic pathway, and it will go through, it's not a pathway, it's just a single step and referred as pyruvate oxidation, where pyruvate or pyruvic acid will be converted to firstly acetate, but then it will combine with coenzyme A, become acetyl coenzyme A. Okay? And by now, the pyruvate already gone into the mitochondria, and then through pyruvate oxidation would have formed acetyl coenzyme A. And then acetyl coenzyme A will enter citric acid cycle, where eventually it will become carbon dioxide. Okay? But that's not the only story. Along the line of glycolytic pathway, pyruvate oxidation, as well as citric acid cycle, what other things will be formed is the reduced coenzyme of NADH here, as well as here, and also in 3 locations of the citric acid cycle to form NADH and also FADH2. Also want to emphasize here, within glycolytic pathway, within citric acid cycle, there will be also substrate level phosphorylation, meaning there will be formation of ATP at intermediate level, as well as here. So that's covering in terms of glycolysis, pyruvate oxidation, citric acid cycle. There is another pathway that I call it as all this as the major catabolic pathway, that is electron transport chain. Within the electron transport chain, which is happen to be found in the mitochondria, all those NADH that has been generated within glycolytic pathway, pyruvate oxidation, as well as FADH2 from the Krebs cycle will now enter electron transport chain, where they will be oxidized to generate NAD plus as well as FAD, and most importantly to become ATP. So that's the complete process or overall in terms of glycolytic, pyruvate oxidation, citric acid cycle, as well as electron transport chain. So we just learned the fate of pyruvate under aerobic condition. What about anaerobic condition, whereby there is not enough of oxygen, or a particular tissue such as erythrocyte or red blood cell which has no mitochondria? So what happens in this situation is that the pyruvate will be converted to lactic acid. So lactate or lactic acid will be generated, and this is a partially oxidized intermediate where there's a lot of energy still found in lactate.